In this lecture, we're going to look at the steps in muscle contraction. But first, let's take a look at this video.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles. An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin-binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction. During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
And so that video illustrates how the muscles contract. Now, what happens during muscle relaxation? Well, the acetylcholinesterase is going to break down the acetylcholine within the synaptic cleft. Remember, anytime you see ACE at the end of a word, it means one of two things. Either it's an enzyme or you're in a hardware store. Well, in this case, since this is anatomy, we're talking about enzymes. So acetylcholinesterase is the enzyme that breaks down acetylcholine into acetic acid and choline. And if you remember, I've talked about before, acetic acid is what? Vinegar. Well, how expensive is vinegar? It's pretty cheap, right? So we don't really need to recycle that, but we're going to recycle the choline. So we reabsorb the choline back into the nerve ending, into that synaptic bulb, and combine it with some more acetic acid. And there we've just made more acetylcholine. And we store those up in vesicles. Okay, so when that happens, let's see, the muscle action potential ceases. Now, calcium release channels then close. As long as there's calcium within the muscle tissue, the muscle is going to still be contracted. So how are we going to get rid of that calcium? Well, through active transport, we're going to pump the calcium back into storage into the sarcoplasmic reticulum. And there we have something called calcium binding protein, or calsequestrin, and that helps to hold calcium in the sarcoplasmic reticulum. And the calcium concentration is going to be 10,000 times higher than in the actual cytosol. And so the tropomyosin troponin complex recovers the binding site on the actin, and so this way myosin cannot bind to the actin and the muscle relaxes and can then be pulled apart by an antagonistic muscle to stretch it back out again. And so let's look at rigor mortis. So rigor mortis is a state of muscular rigidity that begins 3 to 4 hours after death and lasts about 24 hours. Now that's a very broad statement because there's a lot of things that can affect what we call the rigor complex or rigor mortis. One of those things can be temperature. If it's a very hot environment, well, since this is all a chemical reaction, what happens when you apply heat to a chemical reaction? It speeds up. So this can happen a lot quicker. And what happens if it's very cold? Well, think about, a chemical reaction taking place in a freezer. It slows way down. Okay? Matter of fact, if it's cold enough, it may not happen at all. All right? And so again, with rigor mortis, again, on average, with these average temperatures, it's going to begin 3 to 4 hours after death, at which time the body starts to stiffen out and the muscles are very, very rigid. The body is very rigid. After about 24 hours, again, of average temperatures, then the body becomes more flexible and less rigid. So why is that? Well, after death, these calcium ions are going to leak out of the sarcoplasmic reticulum. When it does that, it binds to the troponin, uncovers tropomyosin, and then what happens? The myosin heads bind to the myosin binding sites on the actin. Okay? So we're reattaching, I like to think of them as like magnets. We're reattaching those magnets once again. And what is it that that breaks that bond? Well, ATP. Well, if someone is deceased, do they make ATP anymore? No, they don't. And so as more of these cross bridges happen because of that calcium leaking out of the sarcoplasmic reticulum, that makes the body stiffer and stiffer and stiffer. Now, I said after about 24 hours, the body relaxes again or begins to relax again. And why is that? Well, remember inside the cells, we have lysosomes that contain lysozyme. Well, they start to break down and they release the lysozyme, as well as other proteolytic enzymes that begin to digest the decomposing cell. Okay, and so as the cell starts to break down, because actin and myosin are proteins, as those proteins break down, then they're no longer rigid or stiff, and the body relaxes and becomes more flexible. Let's look at length of muscle fibers. The optimal overlap of thick and thin filaments produces the greatest number of cross bridges and the greatest amount of tension. As you stretch a muscle past optimal length, you have fewer cross bridges exist and less force is produced. And try this sometimes. Stretch your arms out as much as you can and try to pick up a chair, by bending your arms. That chair is going to feel quite heavy. Okay? Now, if the muscle is overly shortened, less than optimal, then fewer cross bridges exist and less force is produced. So try that. Bend your arms all the way, and now bend over and try to pick up a chair. Again, it's going to feel very heavy. And by the way, I don't recommend you actually try these things. You could hurt your back by not properly lifting these things. But that is what's going to happen. Both ways are going to make that chair feel a lot heavier than it really is. And so what happens is the thick filaments are crumpled by the Z-discs. And normally, resting muscle length remains between 70 to 130% of optimum. In other words, if you went to lift that chair again and you had your arms bent slightly, okay, now there's enough of those sliding filaments overlapping to get a good grip and allow you to bend your arm to pick that chair up. Again, it's going to feel way too heavy with your arms straight. It's going to feel way heavy with your arms bent. But with a slight bend in there, you're going to get the maximum strength. And usually during class, when they're in person, I demonstrate with a table. If I go to hold on to just the edge of the table and try to move it, I can't move the table. I can't lift the table because just my fingertips are on the very edge. So I don't have a good grip on it. So that's like having the muscle stretched all the way out. If I lay down on the table and wrap my arms around it and try to lift it, again, I can't get a good grip of it to lift that table up. And so that would be like having the muscles overly shortened. But if I get a good grip of the table and bend my arms a little bit, that's an example of having a bit of contraction or what we would call muscle tone. And that's going to allow me to pick up that table quite easily.